All right, let's go ahead and get started with this last class before the midterm exams. Just one thing that I want to point out, remember that you need to participate in 4 discussion sections in order to get the maximum of 4 points. Many of you are doing great and well on your way. Those of you who are waiting for those last few discussion sections, don't wait. You might get sick and miss one. So go to your discussion sections, make sure you participate, and get that easy point. The last homework, which will cover the material we did on Tuesday as well as tonight's material, that will be posted after class as usual. I will, however, post the key for that at noon tomorrow. You still have until noon on Tuesday to submit for that half credit if you don't make it by 10 a.m. tomorrow. But I'm going to post the key early in case some of you want to look at that key before you take the pre-midterm. And that will open tonight at 5 o'clock. It will be open for 48 hours. It will close exactly at 5 o'clock Pacific time this Saturday. Make sure you have scheduled yourself to take it. Who's going to take it tonight? Who's feeling good and going to take it tonight? Yeah, get it over with. You can do it, and that'll help guide you to know what you need to focus on. Who's going to take it Friday? Good, yeah. Who's going to wait till Saturday? All right, most people are going to take it before then. That's good, but you have until 5 o'clock Saturday. So make sure you do that. And then on Sunday, the TAs are going to hold a review session here in this classroom, and they're going to go over that pre-midterm with you. And that's all in preparation for the actual written midterm, which will be in class here next Tuesday. Any questions? And so for that midterm next Tuesday, the only things you need to bring to class for that are something to write with and your photo ID. Any questions? All right, and this is just to point out so everyone is clear. If you go to the Canvas class website and you look in the exams module, you will see the pre-midterm there. You will be able to open it once it is 5 o'clock. Once you open it, you have to start it, and that's your 80-minute time window. Does everybody know where to find the pre-midterm in Canvas? Awesome. The other thing I want to point out is we have a discussion board in this class. Nobody has ever used it yet, but this is the best place to get your questions answered. If you have a question about a homework, about a clicker question from several classes ago, this gets checked all the time by the IAs and myself. The other great thing about using the discussion board is that the question you have, probably other students have that same question. So therefore, everyone has access to this, sees the question, and sees the answers that we put down. If you have questions about the exam or what's going to be on the exam or anything that we've covered in this class, go ahead and put it on the discussion board, and it will be answered in a timely manner. Any questions? All right. So let's review what we talked about on Tuesday. So the first thing we talked about is this assembly of the pre-initiation complex, this PIC. This is the complex that's going to include those general transcription factors, those TF, Roman numeral 2 for RNA polymerase 2, and then the letters. And this is going to help bring in RNA polymerase 2 to the +1 site so that transcription can start. And what we learned is that there is sequential assembly of these factors, and it's necessary for these factors to come in that order. That's how they recognize the core promoter sequence and bring in the factors in this regulated manner. And I told you an easy mnemonic to use to remember the order in which these general transcription factors come in. And what is that? DNA, bending, to, facilitate, efficient, helicase. Right. And so that means something, right? So TF2D is the first factor or first complex. So when we're talking about these TF2 letters, it is a group of proteins. So TF2D includes the protein TBP and then several additional proteins that we generally refer to as TAFs or TBP-associated factors. So this complex is going to come in first. TBP will be able to bind directly to the TATA box. It is a DNA binding protein that recognizes that sequence and binds directly to it. Then next, TF2A, then 2B comes in, and once that group is there, the DNA gets bent. That allows for Pol2 along with 2F to join the complex. Next comes in 2E, the E inefficient, and finally 2H, which actually does bring in helicase activity to start unwinding that template so that transcription can occur. The rest of the class, we then talked about a variety of different experiments. First, a set of experiments to ask, does a specific protein bind to a specific sequence of DNA? And we talked about two that we consider to be in vitro experiments. What we mean by in vitro is in the test tube, meaning we know exactly what we have put into that test tube. And those two are going to be the DNase 1 footprinting, an electrophoretic mobility shift assay, or just a gel shift assay. And then at the end of class, we started to talk about another experiment that also asks a similar question, is a protein associated with a specific sequence of DNA in vivo, meaning in a cell, in a tissue, in an animal. So you're actually going to cross-link what is happening in that cell type at a given moment or given time in development. And we'll go more into this ChIP assay at the beginning of this class. All right, so looking at the types of results that you can get from these experiments, what experiment was done to see these types of results? The DNase 1 footprinting experiment, exactly. And so the way that you do a DNase 1 footprinting experiment is, first of all, you need radio-labeled double-stranded DNA. And what's very important is that you have the label at just one position, and you know exactly where that position is on your double-stranded DNA. In your control, you're going to put that DNA in a tube and nothing else. The other tube, you're going to put that DNA in, plus the protein that you're testing, whether it binds to. You then treat both samples, both tubes, with DNase. That's the name of the experiment, DNase. DNase is going to go in. It's going to cut everywhere that it can. If there's a protein bound to a sequence, it's going to protect that region from being cut. So you're never going to get a DNA band that was cut in that position. After you've gone through that reaction, you're going to purify the DNA. You're then going to run it on a denaturing gel. So you're going to see all those sizes of bands from where the DNase actually cut your double-stranded DNA. Bands that are closer to the bottom are going to be shorter, and therefore we're closer to where your radio label was. Bands that are bigger run at the top, and they are going to be the longer ones. So they are extending from the radio label all the way to the other end of the DNA. And then you compare your control where you have added no protein to the test where you have added a protein, and you ask, is there any difference? Was there any region that was protected that gives me a so-called footprint? And in this example, yes, indeed, we saw that there was a space here, meaning that the protein bound this DNA and protected it from ever being cut in those positions. So from a DNase 1 footprinting experiment, we get a variety of information. We get one, the answer to that question. Did the protein bind the DNA? If you see a footprint, yes, it bound the DNA. What else can we learn from a DNase 1 footprinting experiment? Sure. Where it bound to the DNA. You know where you radio labeled your double-stranded DNA. And so looking at the position that got protected, you can see, aha, it cut at position 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, whatever. And then you get a space, and then you resume cuts. That tells you, relative where your radio label was, where the protein bound to this piece of DNA. We also then talked about this gel shift assay. So for a gel shift assay, which I think you'll recognize is the results of this experiment here, we also start with double-stranded radio-labeled DNA. One tube, we just have the DNA. The other tube, we add our test protein to. You then take that whole complex and you run it in a non-denaturing gel. What do we mean by non-denaturing? We simply mean we want the DNA to stay double-stranded and we want the protein to remain associated with that double-stranded DNA so that it will migrate differently if it's just the double-stranded DNA versus if a protein bound to it, it's going to migrate much more slowly through the gel. You're going to see this shift, and this shift will then indicate that the protein must have associated with that DNA in order to make it migrate more slowly or shift in the gel. And then finally, we talked about the ChIP assay, which we will go through the steps of that experiment and how to interpret it in a couple slides. Before we talked about the ChIP experiment, we mentioned that we could do then some assays to ask, well, is it important for this protein to actually bind to this sequence of DNA? If that element is there and that protein is there, does it change transcription? Do I get a higher level of transcription, meaning I had an activating or positive expression effect? Or if I get less expression compared to either when the protein's not there or when that element is not there, then that must mean that this protein is able to repress the expression. And so we talked about we could do this either in vitro by setting up our reagents in a test tube and looking if RNA was made, how much RNA was made, or we could make a reporter system and put it into cells. Does anyone have any questions about these types of experiments? Yes? You want to go through a shift? Okay, so when you have just the double-stranded DNA alone, it can run fast through the gel, right? So it's going to be just the double-stranded DNA, and it will run to the bottom of your gel. If that double-stranded DNA is bound by a protein or a group of proteins, it's going to run a lot more slowly through the gel. So instead, it will only get to this position compared to that position run at the same time with the double-stranded DNA. Bigger complexes move more slowly through these non-denaturing gels. And so if I see a shift, a change in where the DNA alone is running, that allows me to conclude that the protein must be bound to that DNA to cause it run at a different position. And remember, it is the DNA that is radiolabeled here. Protein isn't labeled. When you aren't seeing protein, we are seeing the effect of the protein on the migration of the DNA in this gel. Does that make sense? Sure. Yes?
Can you explain again how to interpret the ChIP result?
We're going to go through the ChIP in detail in a couple slides. Anything else on this? All right, let's do a clicker experiment. So to the right are the results of an in vivo assay to test if proteins A, B, or C regulate transcription through element X. So a reporter that has the element versus one that doesn't. Based on these results, where as always higher number means higher expression, which of the following are supported by the data? 10 more seconds. All right, final answers. All right, so a lot of love for answer D. Let's go through this and make sure we understand how to interpret these types of data. Who wants to volunteer their answer and how they got to it? Sure.
I think that's when there's like no protein, was like a 2GFP.
So which answer are you?
My answer is D.
Your answer is D? Mm-hmm.
I think that's with no protein, the answer, the GFP was 2, but with like 14C plus X, there was like 5 GFP, so there was like, there must be some expression in there, positive expression. So that's why I think D is the answer.
Yeah, so it says protein C positively regulates the expression of the reporter through element X. Okay, what do we need to focus on here? We need to focus on our samples that have protein C with or without X, and also compare it to when we don't have the protein with and without X. So what do we see? Well, we see when we have the element and we have C, we have an expression level of 5. When we didn't have the element, when we didn't have the protein, but we had the element, expression level of 2. We have higher expression level, so it is positively regulating the expression. And did it require element X? Well, when we didn't have X, we had no increase in expression. So this is telling us that for C to regulate this reporter, we need element X there. So D, in fact, is the correct answer. Let's go through each one of these, though. And just to illustrate, so again, you can think about how this experiment is actually being done. Remember, what you're doing here is you have an expression plasmid, so you are programming the cell to express either protein A, protein B, protein C, or none at all. And then in those same cells, you're going to put in one set your reporter that does have element X, that can produce GFP if it's turned on, or that doesn't have element X, but it also has the same protein coding gene on it. And then you're just looking when you do or do not have the element, and when you have the various different proteins, are you having effects on expression? All right, so let's go through the rest of these and see why they are incorrect. So A said proteins B and C bind directly to element X. Do I know that from this experiment? No. What experiment would you do to answer that question? You could do footprinting or an EMSA, a gel shift. Exactly. Those tell you direct binding. This tells you, is there an effect? Is there a function of these element in the protein? B says protein B activates the reporter through element X. What's wrong with that? It is repressing it, right? We are going down in expression when B is there and when element X is there, compared to when we don't have the protein available. C says element X regulates the reporter independently of proteins A, B, and C. What does that mean? So whether or not you have element X is causing a difference regardless of whether you have A, B, or C there. So which results are you looking at here? The none, right? So when none of these proteins are there, am I seeing a difference, plus or minus X? Nope. And that's an important control because remember, you're putting this inside a cell. That cell could maybe already be expressing protein C or expressing something else that will recognize it. So that's an important control to do. And we do not see regulation independent of that. So C is not correct. And finally, E says protein A does not bind element X. Well, we look up here, protein A, when we have that, there's no effect compared to none. But does that tell us that A does not bind? No, this experiment does not tell you binding. It could still bind, but just have no effect on expression in this particular system. Any questions about this? All right, so now let's go on to where we pick up where we left off in class on Tuesday, where we started to talk about what we consider to be more an in vivo experiment to look at interaction of a protein with a specific sequence of DNA. And this we call a ChIP experiment for chromatin immunoprecipitation. Excuse me. So what you're doing in this experiment is you're sort of freezing the cell and whatever is happening in the nucleus at that time to identify at this particular time point in response to this hormone, in response to a viral infection, what was happening at this promoter sequence, for example. And so the first thing you're going to do is you're going to take your sample, maybe it's cells, maybe it's tissue, maybe it's a whole organism, and you're going to do a cross-linking reaction. And that is either adding a chemical or using UV to cause covalent bonds to form between proteins and closely associated DNA sequences. So you have everything sort of stuck together. You're going to isolate the chromatin, and then you're going to break it into pieces, right? Because remember, your chromosomes are millions of base pairs long. All the proteins been cross-linked to it. These are huge, huge complexes. And then we use mechanical force, or you can use enzymes to break that chromatin into smaller chunks. Typically, this will leave you with a few hundred base pairs of DNA that is still attached, covalently bonded to whatever proteins it was associated with. And then the next step, this is where the immuno comes in the name of this assay. We're going to use an antibody. And as we mentioned on Tuesday, antibodies are great because they can be very, very specific. They bind to specific proteins. In fact, you can develop an antibody that's so specific that it will detect the difference between histone 3 acetylated on lysine 9 versus histone 3 acetylated on the methyl group. So one antibody will only pull out the lysine 9 acetylated version. Another one will only pull out the lysine 9 methylated version. And another one will only pull out the unmodified version. So antibodies are very specific. And where we use them here is this is what we're going to use for the protein we're interested in. And in this particular case, they are interested in finding if their sequence was associated with, let's say, histone 3 acetylated on lysine 9. So you use antibodies, which are depicted by these blue Y things, which will go in and they will bind only to that modified version of histone 3 and anything that's still attached to it. So at this point, all your DNA is still covalently cross-linked to that protein. You pull out everything bound to that antibody, wash everything else away. Everything that didn't have these green, which is indicating that acetylated version, all the orange ones, those all get washed away. You've now selected for that protein of interest and the DNA that was still attached to it. Final steps are going to be releasing the DNA, which is purifying it, so reversing the cross-links, getting rid of all the protein. And then with that pool of DNA, that selected DNA, you're simply going to ask, was my gene of interest, was that present or not? And the way you can do that is by doing a PCR reaction. You design specific primers to the gene, to the region you're interested in, and you perform a reaction. If the gene you're interested in was immunoprecipitated, there's template there for the PCR to amplify and get a signal. If the gene you're interested in was not associated with the acetylated version of histone 3 here, that DNA sequence would not have been immunoprecipitated. You do your PCR reaction, there's nothing for the primers to bind to. There's no template, you'll get a blank. There will be no amplification. Any questions about these steps? All right, so now let's look at what the results can look like. And so here, what looks to be is a very complicated result, but we're going to go through it and you'll see that it's very simple to understand. So what the researchers here wanted to do is they wanted to analyze a variety of different histone modifications at the interferon beta promoter after viral infection. So let's zoom in on just a few of these panels to make it simpler first. So what did the scientists do? So they had cells, and then they infected them with the virus, and then cross-linked those cells at different time points after viral infection. So at the 0 hour, these are cells where no virus has been added, cross-link those cells. Add virus, 1 hour later, cross-link those cells. Another population of the virus infected cells 2 hours later, cross-link those cells, and so forth. So you're looking at this whole time frame of infection, and each one's going to be a different set that has been cross-linked. All right, so now that you have your cross-linked cells, you're going to isolate the chromatin, you're going to break it into those smaller pieces, and then you're going to use your antibody to ask, was a particular protein associated with the interferon beta promoter sequence? And the first example is shown here. This little alpha stands for anti, meaning antibody. So an antibody against TBP. What's TBP? TATA binding protein, exactly. So they had an antibody against TATA binding protein. And in every single one of these tubes where they had isolated, cross-linked and isolated those cells, broke them into pieces, they're going to do an immunoprecipitation with TBP. So 0 hour time point, 1 hour time point, 2 hour time point, and so forth. After doing the immunoprecipitation and washing away anything that didn't bound, they're then going to isolate the DNA and then use their primers. What are the primers designed against? What are we looking for here? Which gene are we asking is associated with TBP? Yeah, IFN beta, interferon beta. This is this only sequence that we're looking at in this entire experiment. So we're going to use primers for the interferon beta sequence, and we're going to do a PCR reaction. We're then going to run that PCR reaction on a gel. If we got amplification, we'll see bands. If we didn't get amplification because there was no template for the interferon beta that immunoprecipitated with TBP at that time point, it'll be blank, right? So if we look at the results here, at what time point does TBP associate with this with interferon beta?
6 hours?
Yes, you see no bands showing up until you see now at 6 hours. That means that interferon beta was associated with this specific promoter by 6 hours after viral infection. And it looks like it stays associated there, maybe even more association at these later time points. But then what happens at the end of this viral infection? The later time points, it's no longer associated. So you can have dynamic gene regulation. Early in viral infection, you start to have to open up that chromatin. Eventually TBP comes in. It's associated. You're going to detect it. And then as your cell responds to the viral infection, you don't need this gene anymore. TBP is going to leave. You're not going to express it anymore. That's what you're interpreting from here. Now let's look at what we mean by these other panels here. So we also have a control that we do in these ChIP experiments that's called the input control. What this is it is going to be the same starting material. So let's say at 0 time point here, we cross-link our uninfected cells, we break that chromatin into pieces, but then we directly purify the DNA. We don't do any selection, we don't do any immunoprecipitation. If we directly purify the DNA, all the genomic DNA should be there. You should have template for any gene you want to look at. You should get a positive PCR reaction. You should see a band. In fact, that is why you see a band in every lane here. And this is just a control to make sure you're using the same amount of cells, the same amount of chromatin came, and it's before you would select for DNA that's specifically associated with a protein. All right, then there's another panel down here that's actually showing you interferon beta messenger RNA. What technique are we using to look at messenger RNA? Yes. A northern blot, exactly. This is not a ChIP experiment. What they have done is a separate experiment where they have taken these cells that are infected at all these different time points, didn't have to cross-link or anything, just took some of those cells, purified the RNA, ran it in a denaturing gel, did the transfer onto nitrocellulose, fixed it there, and designed a probe, so a sequence that was labeled and would base pair to interferon beta messenger RNA to ask, when do we start to see the mRNA be expressed? And not surprisingly, as soon as TBP is there, so when TBP is there, that means the pre-initiation complex is there, you're ready to do transcription, and yes, indeed, that is when you start transcribing the RNA, making the RNA. So don't get confused. This was just a separate experiment to test. Aha, if we see TBP there, are we actually making mRNA? And indeed we are. Any questions about this? All right, now let's look at this big result here, which is not really any different than when we looked at. So what we've looked at so far is TBP when it becomes associated, the control, the input, and then remember this is a northern blot down here. And now all they're doing, the only difference for each one of these panels is which antibody, which protein was being looked at to ask, is this protein, is this modified version of the histone associated with interferon beta promoter sequence at each of these times after viral infection? So the top panel right here, they're using an antibody against the acetylated version of histone 4 on lysine 5 and lysine 8. And what do we see? We see that the interferon beta promoter DNA becomes associated with this modified form of histone 4 at about 3 hours, 4 hours, 5 hours, 6 hours, 8 hours, and then we lose it later in infection. And so each one of these, the only difference in these panels is which antibody was used to look at which protein is associated with this interferon beta promoter sequence. All right, let's go through these questions. So first question, how was this ChIP experiment performed? We just went through the details of that. Is everyone clear on how you would do a ChIP experiment? All right, this seems complicated because there's many, many different samples, but you're just doing the same thing over and over and over again. And then the only thing you're changing is all these different antibodies to look at different proteins. All right, what if I asked you name all the modifications that happen before transcription starts? So how do you know when transcription starts? Sure.
The messenger RNA got activated.
So yeah, so if I show you a northern blot, if you see RNAs coming on, then transcription must have happened. But most of the time, if we're just looking at the results of a ChIP experiment, you're not going to have the messenger RNA there. So what is the other clue you should look for? Good.
The TBP?
The TBP, exactly. And so once you see TBP there, you know the pre-initiation complex there, and you can assume that transcription is going to start at that point. So to answer this question, name all the modifications that happen before transcription starts. What are you going to look for? So TBP is when we know transcription has started. And then which modifications do we have before transcription is started, before TBP associated? Yeah, you showed me with your hand. Oh, yeah, right before. Yeah, before it. So any modifications, meaning we had this modified version of histone 4 associated with the template because we got a positive PCR product. So we can say H4 acetylation on lysine 5 and 8 must have happened before TBP came on. This is another antibody that's very similar, except for it only sees lysine 8 and not also lysine 5. We get a similar result here. What about phosphorylated serine 10 on histone 3? Yep, we're seeing some signal before TBP comes on. So anywhere that you see a signal, that means before TBP, that means that modification happened before. Question?
Does that mean all experiments will include a TBP?
Depending on the question. If I ask you anything about transcription starting, then yes, you would have to see TBP there, or we didn't really talk about any specific proteins that are part of the core. If I said polymerase 2, you could look for that. All right, what about this next question? Which modifications are not essential for transcription initiation? How would you answer that question? Sure.
Same as question 2. If it's already like expressed before the TPP is expressed, then we don't need the TPP basically.
Not if you see it associated before, after, or not at all, right? So TBP was able to associate, but we didn't need acetylation of histone 4 and lysine 16, right? In fact, we never see that happening here. But here, of course, we do see TBP associated. Does that make sense?
So if there's like initiation, like modification that's happening before the transcription starts, they will also need TPP. Like for those who we answered that number 2.
So we know that by 6 hours, TBP can associate with this gene. So anything that we see only associating later or not at all wasn't necessary for TBP to associate with this gene.
Well, right? It was like started before the TBP, but it's also like be associated with TBP.
It could be necessary, or that's why I just say which ones happen before. We don't know if it was necessary for TBP to join, but we definitely can say that these ones were not necessary for TBP to join because TBP is joined. Does that make sense now? Uh-huh. All right, and we already, at what time point does the pre-initiation complex form? Based on this?
6 hours.
Yep. Okay, pre-initiation complex, going to have TBP there, so 6 hours. And then what does the state of histone acetylation prior to viral infection tell you about this promoter? So you'll look, we've looked at a lot of different acetylated versions of histones. And so at the 0 time point, what does this tell you about this promoter, the state of acetylation? Are any of these modifications there in uninfected cells? No. So it means that this gene, this interferon beta gene, is not in a very acetylated form. In fact, it's probably repressed. It is a gene that is responding to viral infection. And one of the ways it's responding is modifiers are coming in, acetylating, opening up that chromatin, so pre-initiation complex can come in. You can start transcribing, making the messenger RNA. Yes?
So are you saying that we can assume that histone acetylation will not happen unless there's like a viral infection in this gene?
What we can say is that this region we're looking at, any of these specific acetyl modifications are not present. All right. Let's do a clicker question on this now. So based on these ChIP results analyzing protein association with interferon beta promoter, which of the following can be concluded? Yes.
Number 12 also a time point.
Let me, sorry. Oh, I don't know if that's good. Yeah, it'll still keep going. But, yeah, basically this is exactly the image you saw on the previous slide. So these are all time points across here. I would move it, but you know the clicker can be finicky, so. Ten more seconds. All right, put in your final answers. All right, a lot of people chose A. Let's go through these. Who wants to share their answer and reasoning? Yes, Lydia. Exactly. It's what we just talked about. And even though it was hidden by the clicker up there, I think all of you recognize this little A I told you stands for antibody. So what's different in each one of these panels is which protein was being analyzed. And the way you analyze a different protein or a different modified version of a protein is by using different antibodies. So let's go through the rest of these and make sure that we understand why they're not correct. So B says different pairs of primers were used to generate these results. Why is that incorrect? Sure.
Because like we're experimenting on the same gene.
Yeah, we're looking at the same gene. We're looking at an interferon beta promoter in every single one of these samples. What is the only thing I would need to change in this experiment if I wanted to ask if these same proteins associated with the interferon gamma promoter during viral infection? Just change the primers exactly. And in fact, once you have that pool, that isolated pool of DNA, you can look at a lot of different genes just by designing different primers. We often do that. So, same set of primers for this experiment. C says acetylation of histone 4K16 is essential for transcription initiation. So we already talked about this on the last slide. We don't see that version of the protein at all. We know transcription already started, so that was not essential. Does that make sense? D says the pre-initiation complex forms at 3 hours. What did we say? At what time point?
6 hours.
6 hours, right. And this is helped by having the northern blot here, but usually you won't have the northern blot. Look for TBP. If TBP is there, pre-initiation complex is there, transcription can start. And then finally, once it is activated, the interferon beta gene continues to be highly expressed at all time points in this experiment. And so we already discussed that also. We can see from the northern blot, the RNA levels are going down by the last time point, and also TBP is disassociating from this promoter. So it does not continue to be highly expressed at all of those viral time points. We all clear on ChIP experiments now? Yes. All right, let's move on to new material. All right, so we're going to be talking a little bit more specifically about these transcription factors that can act as activators or repressors. We sometimes consider these to be modular proteins, meaning they have separate domains that do different things. In general, one domain is going to be a DNA binding domain. It's going to be the sequence of the protein that recognizes a specific sequence in the promoter region of the gene of interest. So this very complicated diagram here, these are all different DNA binding proteins that recognize specific sequences here in all the color code. And many promoters have this, many different proteins recognize many different sequences. Now, the activator or repressor will not just bind to the DNA, but it's going to do something to the expression. And so the other domain it will have will act as an activator or repressor domain. And we'll see how they do that in a moment. And very often, these transcription factors act as dimers, meaning two proteins. If it's two of the same protein, it will be a homodimer. Two different proteins, it would be a heterodimer. And often this allows the two proteins to stably associate to have cooperative binding. All right, so here's again what we mean by activators or repressors functioning as modular proteins. So these are just examples of DNA binding proteins. In this case, they are all activators. The blue balls indicate the DNA binding domain. This will be the sequence that will recognize a specific sequence of DNA. And in green will be the activation domains, and then there's, of course, linker sequences holding them together. And here's an example. So Gal4, its DNA binding domain recognizes this specific sequence of DNA, whereas SP1 is another activating protein, and it will recognize that specific sequence of DNA. Don't memorize these sequences. I would never ask you something like that. I just wanted you to appreciate that there's lots of these activators and repressors that will have specificity based on what sequences are available. And most genes are going to have a lot of different regulatory sequences, and the presence or absence of different repressor or activator proteins will determine, is that gene turned on? How highly is it turned on? So for example, this EGR gene has these binding sites that are bound by these activator proteins, SRF and AP1. And when those proteins are in the cell, they will bind and you will turn on high levels of expression of EGR. But it also has repressor sites that are bound by this repressor protein. And it turns out if all three of these proteins are present in the cell, it completely represses expression of this gene. So different combinations will have different effects. All right, so because we have these combinations of transcription factors, we can also get more complex regulation. We can have factors that dimerize, meaning the two need to come together in order to stably bind and regulate transcription. We can have these two different proteins, let's just say factor A and factor B. And so factor A's DNA binding domain might recognize a pair of sequences that are going to be the same or very similar at this site 1. So two of the same, which we call a homodimer, can come together here. This has two sites for B. These two will come together here. But we can also have instances where proteins were heterodimerized, so two different ones. And for that to happen, we would have to have an A DNA binding site and a B DNA binding site here. And maybe when you have the combination of all of these together on this promoter, you get high levels of expression, whereas you may get much lower levels of expression if only B is bound. So why don't we have B bound over here at site 4, bound up here? There's no A, exactly. So this has a sequence that can only be bound by A. If A protein is not expressed in that cell type, not only is it not going to be bound at its site 1, but it's not going to be able to help the B protein bind to this site 4. And so you could have the protein not expressed. What's another reason why we might not have A being able to bind? Might be there, but yes? An inhibitor protein, right. There might be an inhibitor. It could be a protein. It could be a nutrient. It could be a factor that binds to A and prevents its DNA binding activity. Here's another example of cooperative DNA binding. So at this particular position, there is a weak binding site for this transcription factor NFAT, and it's right next to a weak binding site for AP1. When these two are both present in the cell, though, they will have protein-protein interactions that will allow for cooperative binding to this site so that it can then regulate transcription. What do we mean by a weak binding site? Anyone remember what that actually refers to? Yes? Yeah, exactly. It's not consensus anymore. And so it turns out that in IL-2, so this is a very important immune gene. You need to tightly regulate it. If you overexpress it, you get autoimmune diseases. So in its promoter, it has an NFAT binding site that looks like this sequence and an AP1 binding site that looks like this sequence. Turns out that the consensus, meaning the site that NFAT binds to most strongly or binds to best, actually has an A at these two positions. So it is a non-consensus sequence in this IL-2 gene, which makes it a weak site. Same with AP1. AP1's consensus sequence looks like this. We have a 2 nucleotide change, meaning it's weaker. And so only when you have these proteins both together in the cell are you going to get this cooperative interaction where they can then stabilize each other's binding to this site and allow for high expression of this gene. All right, so let's talk a little bit about the mechanisms of transcriptional repression and activation. So what is actually happening for these proteins that act as activators or act as repressors. And then again, we'll review the types of histone modifications that are more associated with repression versus activation. All right, so what activators and repressors do in a very general sense is they're going to modulate chromatin structure. If you want to activate expression, you want to open the chromatin. If you want to repress expression, you're going to help condense it. And so they are also going to interact with other proteins. And one protein that is, or one big complex of proteins that is recruited by activating proteins is called mediator. Sort of mediates the interaction between the activators and the core general transcription factors. And so if we are talking about a repressor, for a repressor to function, to recognize a specific gene and start to shut down its expression, it needs to have a DNA binding domain. It needs to recognize a specific sequence in that gene. So it's going to use its DNA binding domain. That's what DBD stands for. So this is a protein, UME6, that's going to bind to the DNA binding that is using its DNA binding domain to recognize a specific sequence. So this URS is a sequence in a particular gene that is bound by the DNA binding domain of UME6. It's then going to use its repressor domain to recruit what we call in general co-repressors, other proteins that are going to help shut down expression of that gene. And of course, one type of protein that you would want to recruit are enzymes that are going to modify the chromatin to put it in the condensed form. If we want to condense chromatin, we want to take away those acetyl groups, right? So we want to deacetylate, maybe also bring in methyl transferases, methylate that group so it'll be in the more repressed form. So repressors are going to bring in co-repressors to shut down the availability of that gene for transcription. On the other hand, we can have activators. So activators, again, are going to have a DNA binding domain so that they can recognize a specific sequence of DNA. In this case, we're talking about an activating protein called GCN4. DNA binding domain is binding to a sequence called UAS in this particular gene. And then it's modular. So the other domain in GCN4 is the activator domain. It is going to recruit general co-activators. Some of those co-activators are going to be acetyltransferases. We want to acetylate the histone so that they become looser and open up. And then, as we mentioned, once you're opening up the chromatin to help eventually recruit the pre-initiation complex, these activators can bring in this big mediator complex to help recruit RNA polymerase II and those general transcription factors. All right, so here's a question for you. So here's GCN4, it is our activating protein here. If I did a DNase 1 footprint experiment where I only have GCN4 protein and UAS DNA sequence, will I see a footprint? GCN4 has a DNA binding domain, which means it binds directly to a sequence of DNA. So I have this protein and the DNA sequence it binds to in my test tube. Are they going to bind? Yes, absolutely. And you will see a footprint there. Would I see a gel shift if I just have that DNA in this protein? Yes, I would see a gel shift. How about if I did a ChIP assay, use antibodies against GCN4, will I get a positive PCR product? Yes, absolutely. What about one of its co-activators, GCN5 here? What if I put GCN5 in a tube with UAS sequence and do a footprint experiment? Will I see a footprint? No, right. This does not have a DNA binding domain. It is recruited to this region through protein-protein interactions. I also wouldn't see a gel shift. Would I see a positive ChIP result? Yes, you would. This is in close enough vicinity that it would indeed cross-link to this region. So this is one of the reasons I've tried to be careful when I'm talking about the ChIP experiment to not say binding to the sequence and instead saying associating with the sequence or interacting with the sequence. When you do that cross-linking, all these factors will indeed form covalent bonds that are close enough to the sequence. You immunoprecipitate, you're pulling out whatever chromatin chunk had GCN5, and then you're releasing and you're using PCR to look at the DNA. This would show up positive in a ChIP experiment. So that's why if you do a ChIP experiment, you get a positive result and you say, aha, GCN5 associates with this sequence. But then you want to ask, does it directly bind to this DNA sequence? Then you would do your footprint or your EMSA assay to ask that question. Yes?
Will you still need GCN4 to then see it in the ChIP assay?
Yeah, if you had a mutation or deleted GCN4 from the genome, this would not interact with anything. It needs to be brought in through these protein-protein interactions. Good question. Everyone clear on that? All right, so now let's talk a little bit about regulation of transcription factor activity. So for a transcription factor to do something, its expression level is going to be important. Some cells will express some transcription factors, others will not. Even if the transcription factor is there, it can be regulated by the presence of ligands. An example of this when we talked about the lac operon was, of course, lac repressor and lactose. Lactose is a ligand. It is a molecule that interacts with the lac repressor protein. When it binds to lac repressor protein, it changes its ability to bind to DNA. So that is an example of a ligand. And the examples we'll talk about in this lecture are going to be hormones that bind to a type of transcription factor called nuclear hormone receptor proteins. All right, so just to reiterate, in thinking about how cells can transcribe different genes, one difference is going to be whether the transcription factors are expressed in a certain cell type or not. For example, this transthyretin gene is only expressed in the liver. Turns out that it has multiple regulatory sequences, promoter proximal elements, enhancer sequences, and many of these regions are bound by transcription factors, activators that are expressed in many different cell types. But there are a few positions that are essential to be bound in order to get expression, and those sites are bound by HNF1 or HNF3, which are transcription factors only produced in liver cells, only produced in hepatocytes. So even though these cells are general, these proteins are general, these are specific, and that will determine that this gene can only be expressed in levels. So the expression of transcription factors will be an important component. And then the activity. And so again, the protein might be there, but if there's an inhibitor around binding to it, blocking its DNA binding ability, it's not going to be able to regulate transcription. Furthermore, there can be specific molecules like hormones. Hormones are a type of ligand that can bind to their receptor protein to specific transcription factors and regulate their ability to regulate transcription. And that's what we're going to talk about now. So, nuclear hormone receptors are just a type of transcription factor. And as a type of transcription factor, they indeed have a DNA binding domain. So they're going to recognize a specific sequence, be able to bind directly to it. And then they also have a ligand or hormone binding domain. And whether or not the hormone is present and bound to that ligand will determine whether the transcription factor, this nuclear hormone receptor, can activate transcription or not. And then here's just this variety of examples of nuclear hormone receptors that respond to hormones as you would expect, like estrogen, thyroxine, vitamin A for retinoic acid receptor, etc. All right, so these nuclear hormone receptors, there's two general types based on the way they function. They are either functioning as a homodimer, two of exactly the same nuclear hormone receptor together, or a heterodimer, two different flavors of nuclear hormone receptor. looking at this glucocorticoid receptor protein, if I tell you that its DNA binding domain specifically binds to the sequence 5' AGAACA, is it going to function as a homodimer, meaning another molecule of glucocorticoid receptor is also going to bind in this sequence, or is it going to function as a heterodimer with some other nuclear hormone receptor? So if it's the same protein, it has to use the same DNA binding domain to bind to the same sequence, reading it 5' to 3'. Can this bind another molecule of glucocorticoid receptor so that it is reading 5' AGAACA? Yes. So here we're also reading 5' to 3', and so indeed the glucocorticoid receptor functions as a homodimer. Two molecules of the same nuclear hormone receptor, both using their same DNA binding domain to read this sequence. Bind to this region, it is called a response element. This would be the glucocorticoid receptor response element. So you would see this exact sequence, a space of 3 nucleotides that can be anything, and then the same exact sequence over here, although we're reading it 5' to 3' in this direction. All right, so let's look at how nuclear hormone receptors function if they are of the homodimeric type, meaning two of exactly the same nuclear hormone receptor are going to bind a response element and regulate expression. Question? It can vary depending on which ones it is, yeah. And the DNA binding sequence also will be different for different ones. All right, so how are we going to remember the differences between homodimeric and heterodimeric? So homodimeric, that's easy. We know that's two of the same exact nuclear hormone receptor. But it also turns out that this type of nuclear hormone receptor is stuck in the cytoplasm if it is not bound to hormone. So I remember this because homodimeric has 2 O's, cytoplasmic has an O. If you remember that, you'll never get confused, all right? So the homodimeric are going to be stuck in the cytoplasm until the ligand, which is going to be a hormone, comes into the cell, binds to the ligand binding domain. That kicks off the inhibitor that was keeping it in the cytoplasm. It's going to go into the nucleus. It's going to bind a homodimer, so 2 copies are going to bind to the response elements, and it is going to activate transcription. So it's going to recruit co-activators. It's going to modify the chromatin, acetylate it, open it up, bring in the pre-initiation complex, and you will have transcription. Whenever nuclear hormone receptors, whether you're homodimeric or heterodimeric, are bound to their hormone and bound to their response element, they're going to be activating. The homodimeric can only be activated because when there's no hormone, they're stuck out in the cytoplasm, so they're not doing anything to regulate transcription. Now that's different from our heterodimeric nuclear hormone receptors. These are always in the nucleus and always bound to their DNA, to their response element. So again, heterodimeric has these nice E's here, nuclear has an E. That's how we're going to remember it. By heterodimeric, we mean it's 2 different types of nuclear hormone receptors. I'm showing you an example here of the vitamin D receptor forming a heterodimer with RXR. They are in the nucleus. They are always bound to their DNA. And when there's no ligand, when the hormone's not there, they actually repress transcription of the bound gene. Once the hormone comes in, binds to the receptors, they can then activate transcription. So let's look at an example of that. So here we have a response element of RXR and a thyroid hormone receptor bound to the response element of this gene. If there is no ligand, no hormone there, they are bound to co-repressors. What are some co-repressors? They're going to deacetylate the chromatin there so that it becomes more condensed, bring in other types of repressor proteins until hormones come in. The hormones, these ligands, so retinoic acid, thyroid hormone, can come in, bind to the ligand binding domain. When that happens, the co-repressors get kicked off. Co-activators, such as enzymes that are going to acetylate the chromatin in that position, come in, and you activate transcription. Any questions about differences between homodimers and heterodimers? Yes?
I'm a bit lost. I don't see any difference between the hetero and the homodimer. It's like the sequence, they were the same sequence.
So don't worry about the sequence, but the main point is heterodimers means two different proteins that are coming in. Yes, this sequence is the same here, but that's fine. They have different ligand binding domains. The actual sequence of the protein is different. They are functioning as a heterodimer. They are in the nucleus, and they are associated with the DNA repressing. Homodimers stuck out in the cytoplasm. They're not bound to any DNA until the hormone comes in, and then they can go into the nucleus. All right, let's do a clicker question. So to the right are the results of a ChIP assay to analyze the nuclear hormone receptor PPAR-gamma and other proteins associated with gene X in the absence or presence of 3 different ligands. Which of the following are supported by the data? All right, 10 more seconds. All right, put in your final answers. Oh. Okay. Let's go through this. Make sure we understand how to interpret a ChIP assay and putting together what we just learned about nuclear hormone receptors. Does anybody want to share their answer? Probably E, right? Probably more than one of the above since we got multiple answers. So let's go through each one and see if there were multiple answers. A says PPAR-gamma functions as a heterodimer. What would we look at to think about that? What do we know about heterodimers? Yes? They'll not only be in the nucleus, but what will they be? They will be bound to the response element. They will be bound to the DNA sequence. If you do a ChIP, are you going to find that protein there? PPAR-gamma. Is it associated with the gene regardless of whether there's ligand or any hormones there? Sure is. Is it acting as a heterodimeric nuclear hormone receptor? Yes. Yes. They're bound to DNA all the time. If you see it there all the time, must be acting as a heterodimer. If you didn't see it there all the time, would be a homodimer, right? Some of the times it's stuck in the cytoplasm. So A is correct. B, all three ligands bind and regulate the activity of PPAR-gamma. When a hormone or a ligand binds to a nuclear hormone receptor, what happens? It activates. And what is one of the things that helps activate the gene? Acetylation, exactly. So let's see. Do all three ligands potentially do something? We look up here. Here is our three different ligands. Are they all causing acetylation? No. Which one is? Right. We only see that happening with C. So from these data, we can say, no, all three ligands do not bind and regulate the activity because we would see acetylation if they bound. So B is not correct. C says SMRT is a partner of PPAR-gamma required for cooperative binding to gene X. So here we have this SMRT. It's there. Oh, we lose it with C. Was it necessary for PPAR-gamma to always be associated with this DNA? No, because PPAR-gamma is still associated even when SMRT is not. In fact, SMRT is a co-repressor, if you think about what's going on here, right? When it leaves, we get acetylation. All right, so C is not correct. D, co-activators modify the chromatin at gene X in the presence of ligand C. Did our chromatin get modified when we have C? Yes, we went from a repressed state where we were methylated to now being acetylated in the presence of C. So yes, the chromatin was modified in the presence of C. So was more than one correct? And it was A and D. Everyone see that? All right, so you are now ready for your exams. We've learned everything new that we're going to learn, and so I just want to give you a few tips. So we talked about a number of different molecular methods, and a good way to make sure you understand what you need about these is to think about what are the experimental steps and what can I and can I not learn from these experiments. Remember, we just talked about the ChIP assay. That's great for telling me if a protein is associated with a gene or a sequence of interest, but it doesn't tell me direct binding. I would need a footprint or an EMSA to do that sort of thing. So I highly recommend you know all these different experiments and think hard about what you can interpret from each one of those. And here's an example of an exercise we can go through with this. So how would you test the following? So how would you test the general chromatin structure of this TTR gene in hepatocytes versus fibroblasts? Which experiment? General chromatin structure. DNase protection, that very first experiment we talked about. The DNA is protected. You don't chop it up. You will detect it. If it was open and loose, euchromatic, you cut it up in that DNase protection experiment. There's nothing for your probe to bind to. How would you tell if HNF1 associates with the TTR promoter in liver tumor cells? ChIP, exactly. Notice I use this word associates, right? So ChIP is when we're looking in vivo, something that is happening in these cells, not just what I put in a test tube. So ChIP would be the best experiment. How would I determine the size of the mRNA expressed from this gene? The size of the mRNA. Northern blot. Northern blot. We are looking at RNA. Northern blots tell us size and how much. How would we determine which promoter sequences are required for transcription? Which promoter sequences are required for transcription? Footprints tell us binding. We want to know, is it transcribed? Is this promoter able to drive transcription? A reporter assay, right? And then I could use deletion analysis or mutational analysis to find the regulatory sequences. How would I find the specific DNA sequences bound by these HNF activators? There's our footprint, yes. So DNA binding protein HNF, which sequences in the DNA is it going to bind to? What if I wanted to determine if there's sequential binding of the activators, meaning does HNF1 need to bind before 3? I already heard the answer. EMSA is best for determining that sequential binding of factors, seeing a bigger and bigger shift on our gel. Any final questions? All right, good luck. You still have discussion sections and office hours and things to go to, so take advantage of those and put your questions in the discussion board. We will answer them.